All right, so in this video, I'm going to review the link between muscle and mitochondria. See, we typically learn about these two subjects separately. Most learn about muscle in anatomy class, mitochondria in biochemistry, and then we quietly forget about both of these things until, for whatever reason, they start to matter in our lives, right? Still, even so, the two are rarely talked about together, but they really should be. See, skeletal muscle and mitochondria shape each other continuously. Muscle contraction signals the building and maintenance of mitochondria inside the muscle. Mitochondrial capacity, in turn, determines what the fiber can do, how it handles fuel, and through signaling pathways that have nothing to do with ATP, what kind of fiber it actually becomes. So today's lesson, we're going to cover a few concepts here, right? First, we're going to discuss why muscle matters for mitochondria. Then we're going to cover a few things mitochondria do beyond ATP. Why muscle is a metabolic organ, how contraction acts as a signal, and how muscle and mitochondria actually shape each other. Then we're going to end this lesson by showing kind of what happens when we stop exercising as we age. All right, so why muscle matters more, why muscle matters for the mitochondria, right? Now, to be clear, muscle is not the most dense in terms of mitochondria, right? Cardiac muscle, the liver, brown adipose tissue, these things are all denser in mitochondria per gram than the skeletal muscle is. But still, if you think about it, skeletal muscle is roughly 40% of the body weight, in a lean adult. So even at moderate density, it represents a big mitochondrial pool. Now, the second thing, and more important in my opinion, is plasticity. See, the mitochondrial content in muscle isn't fixed. It actually responds to demand, right? To a degree, unlike any other organ that we have. So muscle, the other thing about it is it's heterogeneous. So we have type 1 fibers, and these are oxidative, mitochondrial rich with dense capillaries, and they are resistant to fatigue. Then we have type 2X fibers, and they're glycolytic, and they have poor mitochondria. Then we have type 2A, which kind of sits in between them, and this is going to be one of the most adaptable versions. Now, let's talk a little bit about mitochondria and the things they do beyond ATP, right? I think everybody knows by now that mitochondria create ATP through oxidative phosphorylation, but they do so much more than just make ATP, right? We'll briefly go through 4 of these functions right now. The first thing, fuel selection. All right, so mitochondria are where fat and glucose actually get burned. But what gets burned at any given moment isn't decided by the mitochondria alone, right? We have things like hormone, how much fuel is going to be delivered, how much energy the cell is demanding. All of these things kind of feed into that. But the term metabolic flexibility, right? Metabolic flexibility is the ability to switch between these fuels appropriately, depending on whether you just ate, maybe you're fasted, or maybe you went out for a run, right? Being able to switch between glucose and fatty acids. Now, muscle is going to be a major contributor to that metabolic flexibility, and metabolic flexibility, or inflexibility, I should say, shows up in people who have insulin resistance and type 2 diabetes. All right, the next thing that we need to know about is redox signaling. See, reactive oxygen species are going to be generated at the electron transport chain as a normal byproduct of just respiration. Now, at physiological levels, they also function as signals, and they're among the most upstream inputs that actually drive adaptation to exercise. Then we have calcium handling. All right, so the sarcoplasmic reticulum does most of the work here, but mitochondria are also involved. So the sarcoplasmic reticulum releases calcium to trigger a contraction, then pulls it back in. But the mitochondria actually take up some of that calcium, and they read it as a demand signal. Calcium going up means that work is happening, which then tells the mitochondria to increase the output. Okay, the last thing that we should mention is that mitochondria are dynamic. They are built, they're degraded through mitophagy, and they're continuously reshaped through fusion and fission. Therefore, the amounts of and the quality, I should say, of mitochondria present are always going to be changing. All right, now let's shift gears a little bit. All right, let's talk about why muscle should be considered a metabolic organ. See, muscle is more than just aesthetics, okay? It's an entire organ. It's an endocrine organ. It's a metabolic organ. And if you look at, these hyperinsulinemic, euglycemic clamps conditions, then this is, studies, right? 80 to 90% of infused glucose is taken up by skeletal muscle. This is why insulin sensitivity in practice is largely a measure of muscle quality, and the amounts. But there are two main physiological states that we need to know. First, we have the postprandial state after we eat, and we have the fasting state before we eat. Okay, so postprandial and insulin stimulated disposal is going to be a predominantly muscle phenomenon. Fasting glucose, on the other hand, is largely going to be influenced by hepatic, it's a hepatic phenomenon, right? Governed by glucose output and gluconeogenesis, the creation of glucose. Now, muscle is also a major site of that fat oxidation at rest, but it's the dominant site during activity. The part that many people forget, or maybe perhaps never even knew, is that muscle also functions as an endocrine organ. See, contracting muscle releases something known as myokines. And these myokines, they signal the liver, adipose tissue, bone, and brain. Interleukin-6 is a myokine. It's one of the most well-known, and it's going to be released acutely during contraction, right? When I say contraction, I mean contraction of muscle, okay? And interleukin-6 has metabolic effects that are distinct from those that are seen with chronically elevated interleukin-6 from inflammatory states. In other words, when muscle secretes interleukin-6 acutely, right, in a response to exercise, it's going to be rapidly clear and it might be beneficial for the body. Now, conversely, chronically elevated interleukin-6 during inflammatory states comes mostly from adipose tissue and immune cells, and that chronic elevation from adipose and immune cells actually impacts our health quite differently. So let's talk now about contraction. We brought that up, right? Contraction of muscle. Contraction is a signal. So in 1967, John Halozzi reported that endurance training in rats roughly doubled mitochondrial enzyme activity in skeletal muscle. Now, when we look at human studies, right, human training studies commonly produce increases of about 40 to 60% in terms of markers of mitochondrial content over the course of 6 to 12 weeks. Now, it's not double like the rat studies show, but it's still a lot. So how does contraction turn into more mitochondria? See, every time a fiber contracts, two things happen inside of it. Calcium, number one, floods the cell, right? And then number two, we have increased turnover of ATP. Now, even though we're using up and churning over ATP more, ATP levels actually barely drop. The cell is really defending those levels. What changes, though, are going to be the byproducts. So ADP and AMP, these things rise. Simultaneously, phosphocreatine decreases, right? So phosphocreatine falls. Those are the signals that energy demand has gone up. And the cell has sensors for both of these inputs. So calcium is going to activate enzymes called CAMK and calcineurin. And the shift in energy status activates something known as AMPK. Now, both signals are going to affect a protein called PGC1 alpha. You can think of PGC1 alpha as a coordinator, right? Switch it on, and it turns on the genes that actually build mitochondrial machinery. That includes factors like TFAM, which travels to the mitochondria and drives their own genes, right? Mitochondria carry their own DNA. So building them means coordinating kind of two separate sets of instructions at the same time. We have the cells set of DNA, and then we have the mitochondria's DNA. So PGC1 alpha is the thing that kind of keeps these things in sync. Now, if we zoom out, right? Zoom out from the cellular lens for a second and look at this from a practical point of view, two things can happen. So in one 6 weeks endurance study, mitochondrial volume actually increased by 55%. But the number of mitochondria actually didn't change at all. The existing mitochondria simply got bigger. Now, other protocols and other studies do appear to change mitochondrial number and the structure of the cristae itself. So in other words, what am I saying? In other words, mitochondria might increase in number or get bigger, right? So a better way to think of mitochondrial biogenesis is as a remodeling and expansion of a network rather than simply just, oh, they're making more mitochondria, right? There's also some evidence that resistance training appears to improve mitochondrial respiratory function. So Porter and colleagues found increased maximal coupled respiration after 12 weeks of high load resistance training with only a modest but non-significant rise in something called citrate synthase. This is a marker that we use to kind of estimate how much mitochondria we have. Now, what does this imply? Well, this implies improved ATP producing capacity independent of how many mitochondria we actually have. See, mitochondria shape what muscle is and what it does. So muscle contraction impacts mitochondria, and the mitochondria also shape the muscle. So let's talk a little bit about the athlete's paradox as an example. All right, so Goodpasture and colleagues found that endurance-trained athletes actually carry what's called intramyocellular lipids. And this is just fat deposition inside the muscle, right? And they have this lipid deposition well above those who are lean and sedentary controls and in the same range as patients with type 2 diabetes. The difference is that even though athletes have this, the athletes are insulin sensitive. The distinguishing feature here is that athletes have high oxidative capacity. Now, other studies have found similar conclusions, but the point is that lipid content fails to predict insulin sensitivity once we start to incorporate training. Once training enters the picture, the lipid deposition changes. So total lipid inside the muscle isn't necessarily pathogenic, all by itself. What seems to matter is the lipid species that are present, where they're going to be stored, how fast they turn over, and whether the supply is outpacing its use. So greater oxidative capacity supports better lipid handling. Now, when Dubé and colleagues trained previously sedentary, obese, older adults, right, for about 16 weeks, they noticed that intramyocellular lipids increased 21%. But diacylglycerols fell by 29%, ceramides fell by 24%, and insulin sensitivity actually improved. Okay? Now, intact oxidative capacity keeps lipids moving through the mitochondria. Impaired capacity allows diacylglycerols and ceramide species to actually accumulate and interfere with this insulin signaling. Now, one of the strongest human studies actually linking inactivity, oxidative capacity, and insulin resistance used a very simple design where they immobilized one leg and left the other leg alone for every single participant. So Belay and colleagues actually braced 10 young men so that they couldn't bear weight on one leg, and they did this for 12 days. Then they challenged both legs with a lipid infusion to see how each leg actually handled the load of fat. Then they compared both legs and they noticed something. They noticed that the leg that was suspended, that wasn't moving or that wasn't bearing weight, actually had lower mitochondrial oxidative capacity. They had higher intramyocellular lipids and impaired insulin signaling. So here we have the same person, the same diet, the same genome, right? And the same amount of muscle mass. The only variable, the big difference here was that it was 12 days of inactivity on one leg. And that 12 days of inactivity was enough to actually change how a muscle handled lipids. Now, it doesn't prove that mitochondrial decline alone cause insulin resistance, but inactivity, right? Because inactivity changes a lot of things at the same time. But still, what's interesting is that the inactivity for just 12 days was clearly a factor that drove the changes that we actually care about. Now, everything that we've kind of talked about so far, we've talked a lot about fuel handling, but mitochondria do something else that's super interesting. They influence what kind of fibers you actually have, what kind of muscle fibers you actually have, and whether the fiber actually survives or gets degraded. Remember PGC1 alpha, we spoke about this briefly. It's the coordinator that contraction will switch on or off, or I should say it switches on, right, once you contract. So researchers forced it on in those fast glycolytic fibers of mice. Those fibers, if you remember, have almost no mitochondria. Now, after PGC1 alpha was turned on, those fibers shifted towards the slow oxidative type, and they actually became resistant to fatigue. So that one signaling protein changed the fiber's identity. The second finding is about survival. See, muscle has a built-in demolition program. We have a protein called FOXO3 that switches on two enzymes that actually tag muscle for protein breakdown. That's the process that actually occurs when we see muscle wasting in situations like starvation or bed rest. But PGC1 alpha suppresses the FOXO3. So if we exercise, we turn down that whole demolition program. So the same signal that actually builds mitochondria also defends the fiber that actually houses the mitochondria. Now, to be clear, these findings came from mouse studies, but still fascinating nonetheless. All right, so what happens when we stop exercising? So in one study, 7 days of bed rest in healthy young men actually reduced mitochondrial DNA content by 15% and 3-hydroxylacyl-CoA dehydrogenase activity by 8%. And it abolished the normal exercise-induced rise in AMPK, phosphorylation, and PGC1 alpha mRNA. So inactivity doesn't merely lower capacity, it actually degrades the muscle's ability to respond to the stimulus that would actually restore it. Mitochondrial respiration also decreases, okay? So 21 days of bed rest produce a global decrease in respiration in one study, and insulin sensitivity is going to deteriorate faster than the mitochondrial capacity does. So there's proteomic work that says that PGC1 alpha downregulation and redox imbalance appear after just 8 days. Now, these findings matter when we consider how many people are sedentary or confined to bed rest for various reasons, right? Perhaps we're talking about post-surgical procedures, maybe during pregnancy, or maybe even after an injury. Now, shift gears a little bit and talk about aging. Aging is going to be kind of the harder question to answer. For years, study after study reported that older muscle actually had lower mitochondrial ATP production, less mitochondrial DNA, and reduced activity of these key metabolic enzymes. But then something unusual happened. One of the labs that actually produced those original findings, they went back and they looked again many, many years later using similar methods and the similar population. This time, they actually found no age-related impairment at all. So mitochondrial capacity in the older adults actually was intact. So then, which is it, right? Is there age-related decline or not? And part of the answer seems to be about who's being studied. See, there appears to be an age-related decline in mitochondrial ATP production and citrate synthase activity in sedentary adults. Now, what's interesting is that these findings are going to be largely absent in those individuals and older adults who are consistently performing endurance training. So a lot of what gets called aging might actually just be something known as disuse. We don't use it. So muscle gets used less as people get older for a whole, for all sorts of reasons that have nothing to do with biology. So what does that mean? Less use means less capacity. Less capacity means even more disuse. Over a long enough time horizon, that cycle snowballs into frailty. Right? To be clear, training doesn't erase everything. When researchers compared older adults who had trained consistently for years against younger trained adults, mitochondrial ATP production, like I said, was preserved, but there were other markers that weren't. Mitochondrial DNA abundance and several of the transcription factors that actually regulate the mitochondrial building process were still lower in the older group, despite there being decades of training. So there's still something that happens with age that exercise just doesn't fix. But this means that exercise doesn't stop the decline indefinitely. What it appears to do is push that decline later in life, possibly much later than most people kind of even assume is possible. This is why it's so important just to keep moving throughout the day, engage in resistance training 2 to 3 times per week, increase cardiorespiratory fitness by increasing the heart rate throughout the week. As they say, if you don't use it, you lose it. Eventually, we're going to lose it. But wouldn't it be nice to just lose it later in life, right? And that's the part that I genuinely find interesting, right? It's not that decline becomes inevitable, but that we might have actually far more say in when that decline starts than we've actually, been told and what we actually believe. So, all right, that's all I have for you guys today. If this was helpful and you want to go deeper into lifestyle and how it can actually impact your health, check out the Lifestyle Intensive. It's a 10-hour program that we just launched. The link is in the description. From here, go out, exercise, and I'll see you in the next video. Take care. Bye.